we know a fair amount about MS susceptibility. From a genetic perspective, we've identified more than 200 genetic variants that contribute to risk, but these have not been found to influence severity. And so to start understanding whether there are genetic contributions to MS severity, we assembled 15,000 MS cases with clinical and genetic data, and we also had a second replication cohort consisting in 9,800 people with MS. We genotyped all of these individuals and had 7.8 million genetic variants for analysis. We focused on a phenotype that is the EDSS adjusted for age, the age-related MS severity score. And we preferentially recruited individuals that were older, on average 52 years. They had about 20 years disease duration, with the notion that they will have effectively declared their long-term outcome. So we first performed a genome-wide association study, and we found a novel genome-wide significant variant that influences MS severity that is not associated with MS susceptibility, and that was successfully replicated in an independent cohort. This variant was also associated with various longitudinal metrics that we've looked at in a subset of 8,000 people with MS across 54,000 visits. And we found, for example, that this variant influenced confirmed disability worsening at 24 weeks. Homozygous carriers of this variant also had a 4-year shorter median time to EDSS 6, so to use of a walking aid. And we've also saw that this variant is associated with MS pathology in the Netherlands Brain Bank, where we were able to look at cortical demyelination rate in brain stem lesions in a large cohort of 290 autopsy brains in people with MS. And we saw that the genetic variant in homozygous carriers nearly doubled the cortical pathology and the brainstem pathology. And so then we've tried to understand what the biology behind this is. We've nominated MS severity genes based on various approaches, and we found that what these genes had in common is that they had very high expression in oligodendrocytes and in oligodendrocyte precursor cells. So that was an interesting finding for us, and we were able to derive similar insights from genetic variants genome-wide, so not just focusing on that genome-wide significant variant, but across the whole genome, we were able to ask, are there particular tissues where we see the strongest association for MS severity? And we did that across 205 tissues in a hypothesis-free manner using gene expression data. And we saw that in contrast to MS susceptibility, where we know that we have a very strong immune signal in lymphoid organs and immune cells, for MS severity, we saw a signal exclusively in the central nervous system across various brain regions in the C1 segment of the cervical spinal cord. And so what this is telling us is that it's, while MS susceptibility arises from a fault in the immune system, for MS severity, it's really resilience in CNS responses that are driving the phenotype, so that explain why someone will do well or poorly. And so we have several additional results that support this. For example, we're able to show that educational attainment has a protective effect using various genetic epidemiology approaches. And taken together, what we hope these findings will allow us to do is understand the biology a little bit better by digging deeper into these genes, and also start to provide some guidance with regards to drug development using these genetic variants.